Thank you for a kind presentation, and also I would like to thank the scientific committee for giving us the opportunity to present our work. So, first, Activen is a Swiss biotech company located in Lausanne, but we have a lab in France.
A little louder.
Sorry. Activen is a Swiss biotech company. Do you hear me well now? Do you hear me well?
Yes.
Okay. So Activen is a Swiss biotech company located in Lausanne, so in Swiss, and we have laboratories in France, in Nantes, and also a subsidiary in Shanghai. Our company, the aim of our company is to develop miniprotein, and this miniprotein is a new range of cosmetic active ingredients. Our DNA is from aesthetic medicine, and we would like really to show you how we develop our miniprotein and what is miniprotein. Miniproteins are by definition small proteins below 50 amino acids. They have protein, they can fold, they are stable, and they are really they are specific to their target. Miniprotein are not peptide because they are found in nature's additives. So our miniprotein are able to penetrate in the skin because they are very small. They have a 3D folding, and they are super stable. More and more miniprotein will come in the development. If you look at the literature research, there is a lot of miniprotein under pharmaceutical development. And now also this word is buzzing in the social media. To show you here some influencers. I present today the genesis of our miniprotein, which is named XEP716. This miniprotein was developed based on our knowledge of skin rejuvenation. This miniprotein is a TGF-beta-like miniprotein. And why we decided to work on TGF-beta? Because on this kind of protein, it's not a TGF-beta, but it's acting like a TGF-beta. This growth factor is known to stimulate fibroblasts to produce collagen type 1, but also hyaluronic acid, and stimulate the new extracellular matrix. We did extensive in vitro studies demonstrating that this miniprotein is able to stimulate collagen type 1 at the same level as TGF-beta. You can see here in this different chart. We did our in vitro studies in comparison to TGF-beta. We demonstrated that we have an increase of procollagen type 1, elastin, and hyaluronic acid. And also, because in our company, we have a quite huge expertise in RNA-seq analysis, we compare the genome expression of TGF-beta when we stimulate fibroblasts with TGF-beta and with miniprotein. We found a quite good profile, similar profile. This is what we can see here in this chart. We have a good correlation of gene expression profile of our miniprotein in comparison to TGF-beta. This is why we say that we have a biomimetic of TGF-beta. We have also demonstrated that this miniprotein is able to act on TGF-beta1 receptor, which is the receptor of TGF-beta growth factor. I will present you some clinical data we have done, and also why we can say that this miniprotein presents anti-aging activities. We did a preclinical first clinical study. I will not go in the detail for this first clinical study, which we have demonstrated that miniprotein is able to reduce wrinkles, improve firmness and elasticity, while also improving skin shininess. But one of our questions was, and we know all that with aging, the skin become less thick. We have a dermis, which is, yes, very impaired, sorry. The epidermis is thinner, and also the dermal epidermal junction become flat, sorry. What we can see here, it's a young skin. We have this kind of wavy dermal epidermal junction. With aging, the dermal epidermal junction become flat. So we decided to demonstrate at the clinical level using a device which is called the confocal. And with this device, we can measure at the clinical level the dermal epidermal junction, and also this dermal epidermal junction can be characterized, and we can estimate if this miniprotein is able to act at this level. But the confocal is only able to measure to have optical section of the skin only until 200 micrometers, while with the ultrasound, we can have a full thickness measurement of the skin. So we can really measure the thickness, the dermal thickness and dermal density of the skin. Here is some just pictures to explain you what we measured exactly with this confocal. It's illustrative pictures of the skin, which is kind of histological cut. But when we do optical cut, this dermal epidermal junction appear like this rounded shape. And if we count this rounded shape, if the dermal epidermal junction is wavy, we have a lot of rounded shape. While with aging, this dermal epidermal junction is flat, and we have less rounded shape. So we can count exactly this rounded shape. To show you what this is, it's an histological pictures, and here it's confocal pictures. And in this area, we can see this kind of edge papillae, which is this white rounded shape. I will give you, I will give you some information about the number of people involved in this study. We enrolled 26 women between 40 to 60-year-old, and these ladies should present wrinkles and also fine lines. And we asked them to apply in their any face either the vehicle, so the formula without the miniprotein, or the miniprotein. What we found in this case, which was the measurement of dermal epidermal junction length, we have an increase. Well, it's not well appearing here. We have an increase of dermal epidermal junction length with the use of this miniprotein, and this effect was statistically significant in comparison to vehicle condition. Also here, we were able to measure also the deposition. I think there is a kind of, sorry. Sorry, I explain here the dermal epidermal junction length. And now here we are on the collagen fiber parameter. Sorry. We measured also this white area, which are the collagen deposition. So with image analysis, we can measure the dermal with collagen deposition. This is in this histogram in green. We can see in the product group an increase of this collagen deposition. While with the vehicle, we have no improvement. We have a steady state, which is expected, and we can demonstrate at the clinical level the new generation of collagen proteins. This is data are from ultrasound. So I explain you that with ultrasound, we were able to demonstrate and to measure the thickness of the skin. And we can see after 1 month an increase of the dermal thickness, which is significantly different with the vehicle group. And also we can see that the vehicle, there is no improvement, which is expected, and there is just a cream without any active ingredient. We use also the 3D reconstruction of ultrasound, and what we can see at the baseline, we have a lot of white space, which show that we have an aged skin. So the skin is less densified. And when we do the 3D reconstruction of ultrasound pictures, we can see clearly that we have less white space. So here we have a full thickness of the dermis, which is reinforced, and we have a new deposition of extracellular matrix. Here we can see an improvement of dermal density, which is significantly different from the vehicle group, which we clearly demonstrate the benefit of this miniprotein. now there is a kind of device which the device is able to estimate the age of our panelists. So there is their civil age, but also, and when we compare at the baseline their age, and we see after 2 months of twice daily application, we can see that the estimated age by image analysis is reduced. So we can estimate an improvement and the rejuvenation of the skin thanks to this device. While with the vehicle condition, we have no improvement, and also quite, we can mention a tendency to increase the age. Just is some picture to show you that depending on the age of the panelist, either we can have a kind of reverse aging process, or we have a kind of slow down aging process. We have less wrinkles which are appearing here, and here we have a reduction of wrinkles. A summary of this first clinical study shows that we have clearly demonstrated an increase of skin elasticity measured by cutometers after only 2 weeks with this miniprotein, also in comparison to vehicle group. Also here we demonstrate the firmness effect measured with cutometer, just a summary. And when we go deeper in each analysis, we notice that 96% of our panelists, when we see there is an improvement, we have an improvement, an increase of this parameter. So we can say almost all people who apply this miniprotein in this control study, we have a benefit. This is the third part of the study, but we would like to go deeper, and because one of we know that for the consumer, one of the first claim which is looking is fine line, wrinkles, and improvement. So we have seen this benefit, but loss of firmness and skin sagging is the second claim which the woman would like to improve on their face. So we launched another clinical study targeting the oval face and this sagging. What we did also in this study, we enrolled 33 women, which is quite a high number of panelists for cosmetic studies. And usually people who work on sagging, they prefer working on 2 groups, one group with vehicle, one group with only the miniprotein. In our case, we would like to, we decided to work on one group, but hemi-face, which is quite difficult to do this type of study. But because we would like to be sure that the improvement is due to our miniprotein and each panelist is his own control, we decided to work on hemi-faces. Although we use this inclusion, in clinical study, the inclusion criteria is quite important. So we enrolled people having a clinical grading of sagging from 3 to 5 in this well-known clinical scale. And we noticed also when we did a competitive analysis, we noticed that most of people don't measure, quantify the sagging improvement. They just show pictures before and after. So we were looking how we can demonstrate that we have an improvement of this sagging by quantification. Another way also to be sure that we don't have an improvement because the woman enrolled in the study lose weight, we measured the BMI, which is body mass index, because usually what we notice in clinical studies, most of ladies, when they start a clinical study, they do diet in parallel. So we would like to be sure that, yes, if we have a benefit, it's not because they lose weight during the study. So I don't show the data from BMI, but we demonstrate that there is no modification on their BMI during the study. And also to calibrate the grading, because we measured, here, we measured this yellow area. In this yellow area was calculated by image analysis, but we were asking us, but yes, if we improve this yellow area, can we be sure that this yellow area, because there is a reduction, it's correlated to a clinical grading? Because we can show data, but it's not relevant. What we did, first we say if our clinical grading based on this famous scale in our hand is correlated to the age. What we see here we have the age of our panelists. It's our own data, and here is the clinical grading done by the center. We can see a correlation between the age and grading, so the center is able to grade correctly. And when we do the correlation between the clinical score and this measurement at the baseline from our own measurement of clinical panelists, we can see a correlation between a clinical grading and this value. So we have a kind of relevance to demonstrate that if we have an improvement of this parameter is not due to it's not by chance that we improve this parameter. Then I show you some results here. After 2 weeks, we call this parameter oval face reshaping. So it's the yellow area measured. What we can see after 2 weeks, we have a reduction of this yellow area, while in the vehicle group, which is in gray, we have no improvement. And after 1 and 2 months, we have an improvement and a significant difference between the both ME faces. And also what we ask, when we ask the ladies, do they see a difference between their left and right face? 70%, the study was double-blinded, so the clinician and also the panelists don't know what product they applied. So 70% of the women noticed a difference between left and right. So we have an improvement of, and when we check what is the improvement, it was due to the product, in fact. But when we did a 3D pictures with a device called LiveVis, because I showed you just before the measurement of this area, we noticed that we have an improvement not only on this small area, but it's on all the lower face of the lower part of the face. What we did is just illustrative here. At the baseline, we use a mask, a gray mask. And after 2 months, we use a white mask. So if there is an improvement of the sagging, if we do the superposition of white and black mask, we should see the black area below, which is the improvement. And what we can see here in this area where we apply the miniprotein, we can see this fine line of gray. And this is all this area which was in fact lifted and where we have this improvement. So we have another way to demonstrate the oval remodeling benefit from this miniprotein. Just to show you here some pictures here to see. So at the baseline, this lady got kind of a face which is not really rounded, it's a square face shape, and after two months we have a kind of lifting effect here, and we can see that all the improvement is in this area. And then just to show you here, and just yes, also some just a visual, you can see here the before and after of this lower face area. We can see in this lady the improvement of this area. Just to conclude, we have shown by using at the clinical rated using advanced methodology, confocal, ultrasound, that this miniprotein is able to rejuvenate dermal epidermal junction, which become in fact more undulated. We show at the clinical level a new deposition of collagen synthesis, but also the densification through the full dermis, and also the oval face restoration. This study was done, we did a lot of studies in comparison to vehicle, which make in fact the study more relevant, I think, and because we have all this kind of control of the study. And just to, for your information, this miniprotein was awarded in prestigious award, for example, for the cosmetic application in CEW. It was in 2023. Innovation for BSB, CNTLE award is also innovation. So we won a lot of, and also cosmetic 360, which is a prize given by Cosmetic 360 Association in France. And we are quite proud of this miniprotein, which give a result very fast. and people, I think, like this type of miniprotein. So I'm happy to take some questions if you have some questions.